I will invite Dr. Hakon to join us here. About Dr. Hakon, thank you so much for joining our webinar.
Yeah, so I'm going to tell you about an extra one program that we have at Children's Hospital in Philadelphia. This is actually quite a unique cohort because we have several hundred children who have both congenital malformations and have suffered from a pediatric cancer, and in one and the same child. And the question is, do they have a single gene defect causing both, or are these two independent factors? And so we are looking sort of at the correlations between these sort of two phenotypes with respect to the genetic underpinnings. Now, as you know, if you have multiple, or if you have a congenital anomaly or malformation, you are predisposed to have cancer. So there's perhaps sort of reasons to believe that there may be overlapping pathways or genetic variants that contribute to both of those. This project is sort of set to look into that. And here is the cohort selection, and I am not going to go over this definitions here in detail, but these are the electronic medical record codes from the ICD-9 and 10 codes that we use to capture congenital anomalies and also to capture cancer. And there were 1,464 probands that we identified, and as you can see on the slide, 832, they overlap between cancer and having a birth defect. Now we sort of had envisioned that this would be a larger overlap, but a number of these individuals had sort of overgrowth syndrome and other benign growth parameters, so we removed them, and they are sort of listed just as birth defects. And a few of these individuals had cancer alone without a birth defect, but the vast majority had both. And there were 141 trios, so we could sort of look at the parents as well. And this is a slide that just shows the age distributions in males and females, and so you can see that the average age is probably around 18 or 19 now, but these are pediatric patients when they were recruited. And then you can see the ethnic background and the race split up between males and females. Now this is sort of dominantly European ancestry individuals, but there are 157 African American males in the study and 123 African American females. And then you see the family structure, as I said, and the broken up trios between males and females. And this is sort of that most people are aware of. I mean, the samples whole genome sequencing came from the Broad, one of the sequencing center, and we sort of drove the phenotypes generating HBO term, and this was sort of scored by Sortan and AWS Comprehend Medical. There was comprehensive analysis for single nucleotide variants as well as copy number variants, and we used programs that we developed largely at our center at the Applied Genomics at CHOP. One is called GD Cross, and this is a variant sort of ranking and prioritization software that sort of gives you pathogenicity and the most likely variant based on the input of the HPO terms, and the copy number variants are analyzed by the PennCNV and sort of the internal programs we have built at CAC. All sort of chromosomal structural variants were visualized for validation, and we looked into similarly mitochondrial sequence analysis, fusion, expansion defects, and so on and so forth. And this sort of just shows the pipeline. There were 1,806 individual totals, other individuals, family members to the 1,400 probands. And you can just sort of see there were two steps really that we, for the data curation and sort of all kinds of tool development and benchmarking that we needed to do. So this GD cross, as I said, really is a powerful tool to pull out variants and rank them with respect to causality. And we could actually validate several of these variants because some of these kids, had actually also clinical molecular diagnosis in the alphanumerical record, although vast majority didn't, but we could sort of randomly validate some of them. And then the downstream analysis and functional validation sort of followed, and this sort of discovery pipeline shows that, we looked at both coding and non-coding region, the burden analysis, and the HBO clustering was sort of critically important for the for the downstream function of the GD cross program. And then we, when we came up with variants known or novel, we looked into gene matcher for, there were other cases reported, and we took other variants into functional analysis if they were novel to establish and validate their sort of causality with respect to these phenotypes. And here we can see sort of a summary of the phenotypes in both in the cancer group and in the HBO terms. And so you can see the type of cancers that these individuals had, most commonly in the central nervous system. Just because of time, I'm not going to go into this in detail, but you can see that, essentially every organ system was involved in terms of that there was a wide variety of cancer types, with nervous system cancers, neurofibromas, and leukemias being the most common. And then the HBO terms in terms of organ affected based on the phenotype input. Similarly, nervous system probably was the most common one, 682, and cardiovascular, musculoskeletal, digestive system, and essentially every organ system was inflicted in some individuals. And here's a summary of the molecular findings. There was actually, a sizable number of individuals with chromosomal significant chromosomal anomalies, aneuploidy, mostly trisomy, and we heard a talk on that here before. There were, few monosomy and Turner syndromes, and then you can see here sort of the type of mutations that were, for the homozygous or hemizygous that accounted for 17%. Frame shift, nonsense, splice sites, and other in-frame variants. Missense was 184 that have been uncovered for today. And then you can see the copy number variation analysis we've done, and the size ranges in terms of the, we've captured copy number variants that are both small as well as large, apart from the whole chromosomal aberrations. Pathway analysis was performed similarly across the birth defects and birth defects and cancer. And this, of course, is always helpful to understand the overlap and the integration of these phenotypes in this sort of venue. And anyone can look at the slides and look at this more carefully afterwards. This is just sort of standard pathway analysis that we performed. One of the things that we were interested in knowing, as I said, was to see if there was a single molecular diagnosis for both the congenital anomaly observed and the cancer. We, have multiple such examples, such as the MNX1 here that causes sacrococcal teratoma, and also anorectal malformations. There's the Wilms tumor gene with nephroblastoma and various sort of, urogenital malformations. And there's the porcine gene, focal dermal hypoplasia, so laryngeal papilloma and actually other sort of one signaling related cancers have been described from this gene, as well as congenital heart defects and microcephaly. And so these are just sort of representative example of observation where the same gene is responsible for both the cancer and the congenital malformation. More often we saw the opposite. There were dual molecular diagnoses. For example, the DICER3 gene, pleural pulmonary blastoma, and TDFB2, the Lloyd-Dietz syndrome, developmental delay, mitral regurg, and alike, and the other one. So this individual had clear distinction between these two types of phenotypes, and this is just sort of scaling that a bit. This is not showing nearly all of the dual findings that we have, but just giving you representative sort of and showing where they are located in terms of the tissue type, the SCN1A, obviously epilepsy gene, and neurofibroma gene causes neurofibromatosis, obviously a tumor that can turn into malignancy. ANCAR-D11 is a gene that causes the KBG syndrome, whereas the TSHC1 is known to cause colon cancer. You have 14q12, which causes Rett syndrome, or deletion in that region causes Rett syndrome. And the PROCAR2 is a gene that causes Kalman syndrome, hypogonadalism, and sort of anomalies around that axis. And MYH6, well-known cardiomyopathy gene, LEMOX-D3, a gene known to cause cancer similarly. And you have the exit, the exocytosis, which also, turns into hereditary conditions and cancers. Well-known cancer genes, KMT, 2D, and certainly P10 and alike. So I'm not going to count through this in every single gene, but you can see that that there are sort of independent pathways in a number of these individuals that are separate causes, but there are also a large number of individuals who have a single cause, which is very interesting. We similarly looked at copy number and structural variations. This is just to sort of show the type of size range of deletions and duplications that we can capture. I guess those slides represent the slides here. They show deletions. There's a 12.8 multi-gene deletion here shown, and you see a 10 kb multi-axon deletion here with the center involved. You have loss of heterozygosity, as you can tell, and there's only one allele present, either the A or the B allele in this region. And then single action deletion, we can get down to that granularity with the whole genome data very easily. And obviously, a single action defect can be sufficient to cause a phenotype. And here's just sort of, this is actually plotting 22q11.2 deletions and duplications. And you can see the signature patterns here. And this is sort of really, allows us to train algorithms almost in an AI type fashions to sort of, screen through the entire data set to come up with all deletions that are, segmented into AB, AD, AC, etc. And you can see a clear evidence of deletions or duplications with specified for 22q. So these, whole genome sequence data are very powerful for these type of analysis. So I wasn't going to go further into this, but this is a very unique data set. As you can tell, HPO terms are really the phenotypic driver, and then the input is into the GD cross, where we sort of, capture variants, annotate those variants, prioritize them with respect to disease risk for causality, The EMR phenotyping, obviously, is critically important, and you can do all kinds of burden analysis and cluster analysis. And I sort of, wasn't going to go into that for the sort of this short presentation. But we have, very accurate copy number variation analysis where we have causal genes captured by PenCMV and sort of, using a program called ParCMV2, which Joe Glessner in our group drove and is currently in press. So, this type of a program just sort of, shows you the capacity of working within the Kids First program. And obviously these extra ones that are funded through this mechanism are highly enabling, and obviously opportunities to subsequently integrate and work together with other groups to expand and scale and grow these type of discoveries that we have. And this is the team that sort of, has worked on this internally. Deborah Watson, Amir, and Xiang, sort of with Joe Glessner leading the effort. And obviously want to thank the NIH and the Kids First program and everyone there for their support for enabling this. So with that, I'll stop. Hopefully we captured a couple of minutes, but we'll take it from there.